When we encounter an unknown species, perhaps during an outbreak, how do we identify it? How do we know what it's capable of? How can we learn how to protect ourselves against it? First, let's back up and talk about how bacteria are named. The science of classifying living beings is called taxonomy, and we've been doing it ever since Swedish botanist Carl von Linné, also called Linnaeus, established a system for classification using taxonomic categories in the 1700s. He wanted to minimize chaos as new species were discovered and provide a structure for defining and recognizing any newly discovered species. In the case of bacteria, we use a binomial or two-name system of nomenclature. The scientific name for any bacteria is always the name of the genus first, which is capitalized, followed by the species name, which begins with a lowercase letter. Both should be italicized. The names of the genus and species have a wide variety of origins. Sometimes they were named after the microbiologist that discovered them. In other cases, the name might be related to how the microbe looks or the disease it causes. Now that we've covered the names, let's talk about how bacteria got sorted into their respective categories in the first place. When it comes to classifying bacteria, it may seem a daunting or even impossible task. However, scientists have developed a system to observe, test, and then categorize bacteria into logical relationships. There are three main types of classification: phenotypic, analytic, and genotypic. Let's talk about what each of these entails and then discuss some categories that bacteria are typically sorted into. First, let's talk about phenotypic characterization, meaning the set of observable characteristics of bacteria. Those are size, shape, and staining characteristics. Before we had the advanced technology we have now, scientists relied on observing microscopic and macroscopic morphologies of bacteria. For example, using Gram staining, a method developed by Hans Christian Gram in 1884, we can determine if bacteria are gram-positive or gram-negative, and thus how much peptidoglycan their cell wall contains. Gram-positive organisms have a thick peptidoglycan wall, retaining lots of crystal violet stain when using this method, and thus appearing a vivid blue under a microscope. Even just separating bacteria into gram-positive versus gram-negative can tell us a lot about how they might behave. Certain microbes have unique staining characteristics, such as the genus Mycobacterium, which can be detected by an acid-fast stain. Another example involves identifying the shape of individual organisms under a microscope, which will be either rods, cocci, curved, or spiral. Zooming out a bit, scientists also look at how bacteria grow on agar in the lab. They look at the colonies of bacteria that grow, taking note of the size, shape, color, and even smell. For instance, Streptococci colonies tend to be smaller in relation to most other types of bacteria, and Serratia marcescens typically appear red when grown at 22 degrees Celsius. We can test for hemolytic properties on blood agar, identifying if the bacteria produce toxic byproducts capable of destroying red blood cells. For example, Streptococcus pyogenes, the causative agent of strep throat, is a gram-positive bacterium that forms long cocci chains and grows as small white hemolytic colonies on blood agar plates. Since it is likely for multiple species to appear similar in these types of tests, these phenotypic characterization methods serve only as a starting point for further investigation. Next, there are tests to determine what biochemical properties the bacteria have, like the ability to ferment specific carbohydrates, what carbon sources they can use for growth, and the presence or absence of different enzymes like lipases, proteases, or nucleases. All of these observations combined can identify with reasonable precision a species of bacteria. These techniques have also been used to subdivide groups of organisms beyond the species level, down to a specific strain. Beyond the species level, down to a specific strain. Doing this by looking at the genetic makeup of the organism, especially in the case of an outbreak, is called biotyping. Many bacteria also possess antigens, which might be a toxin or other substance that triggers an immune response in the body. Grouping bacteria based on these antigens is called serotyping. Using serotyping, scientists can work backwards using antibodies to detect which antigens are present, thus allowing them to narrow down the bacterial possibilities. Serotyping is a powerful tool for classification, especially for those species that are difficult to grow, those that are difficult to test biochemically, or those that need to be identified rapidly, such as during an outbreak. Scientists can also look at which antibiotics bacteria are susceptible to, which is called analyzing their antibiogram patterns. Finally, using phage typing, scientists can assess which bacteriophages bacteria might be susceptible to. Now that we've covered phenotypic classification, let's move on to analytic classification. Analytic classification methods include whole cell lipid analysis, cell wall fatty acid analysis, whole cell protein analysis via mass spectroscopy, and the presence of cellular enzymes via multi-locus enzyme electrophoresis. Analytic classification can be a bit labor-intensive, requiring expensive machines and specialized training. For these reasons, analytic classification is typically done in special laboratories. Finally, the most precise method for classifying bacteria is through genotypic classification. Put simply, this means using bacterial DNA to determine what species or family bacteria might belong to. In the early days, scientists used the ratio of guanine and cytosine to classify bacteria. As technology has progressed, so has our ability to quickly and accurately identify bacteria using DNA. Using DNA-DNA hybridization, scientists measure the degree of genetic similarity among bacterial isolates. Taking this a step further, scientists can extract DNA from an organism and expose it to species-specific molecular probes. If the nucleic acid probe binds to the DNA, then you know you've properly identified the organism. We can also use nucleic acid sequence analysis to compare unknown bacteria with already known sequences that are unique to a genus, species, or subspecies. Additionally, some bacteria carry plasmids, which are small circular DNA strands that replicate independently of the chromosome. And while the genetic makeup of bacteria can vary drastically between species, their ribosomal genes are remarkably well conserved. Scientists routinely use 16S ribosomal RNA sequences to establish taxonomic relationships between prokaryotic strains. That's why in situations such as an outbreak or epidemiological investigation, scientists can use plasmid analysis or ribotyping to quickly identify bacteria. Now, here's the thing. Bacterial classification into families, genera, and species changes all the time, evolving. Generally speaking, however, our classification system is a robust starting point. Using all of these techniques we discussed, we can organize bacteria into categories and predict their pathogenic capabilities. Let's look at some of these categories for medically important bacteria. First, aerobic gram-positive cocci, which can be further subdivided into catalase-positive cocci, which includes the Staphylococcus group of bacteria, and catalase-negative cocci, which includes the Enterococcus and Streptococcus groups. Next, aerobic gram-positive rods, which can be grouped into actinomycetes with cell wall mycolic acids, actinomycetes with no cell wall mycolic acids, and miscellaneous gram-positive rods. Then aerobic gram-negative rods, cocci, and curved rods, which include a wide variety of pathogenic organisms. Additionally, there are anaerobic gram-positive and gram-negative bacteria, which are further grouped by shape, cocci or rods. Now that we've gotten classification out of the way, let's talk about how these bacteria are able to cause damage to the human body.
From amoebas to aardvarks and fungus to fruit trees, what does it mean to classify organisms? Why do we do it, and how is it done? Classifying anything simply means to put it into a group. In terms of biological classification, organisms are classified or grouped with other organisms that they are most closely related to. These small groups are then classified together into larger groups, and so on, until we reach the top level of classification, which places organisms in one of three biological domains: archaea, bacteria, and eukarya. When each organism is classified in this way, it allows scientists to see the relationships between different species and make sense of a hugely diverse array of life. Modern systems of biological classifications, or taxonomy, are based on the work of a pioneering 18th century scientist called Carl Linnaeus. By studying hundreds of organisms, Linnaeus devised a method of grouping them according to their shared physical features. Those with similar physical features could be classified together. The most similar organisms, i.e. those that could breed together and produce fertile offspring, were designated as belonging to the same species and given a Latin species name accordingly. For example, all domestic dog breeds belong to the same species called Canis lupus familiaris. Other members of the dog family that are closely related, but not the same species, will share the higher group name, the genus name, but have a different species name. For example, the grey wolf, which is the closest relative to the domestic dog, is classified as Canis lupus lupus. By using this system, we can classify organisms according to their shared evolutionary history. Linnaeus also created higher and more inclusive taxonomic categories that encompass an increasingly broader range of organisms. The structure of Linnaeus' taxonomic groups can be seen here. You can make up a little rhyme or acronym to remember this, such as keep ponds clean or frogs get sick. By using this system of common groupings, we can develop diagrams that show the interrelatedness of organisms based on their morphological diversity. For example, this diagram of a taxonomic tree shows that humans are more closely related to chimpanzees than they are to gorillas. A common ancestor is represented by each split in the branches of the taxonomic tree. How long ago did humans and gorillas diverge from an evolutionary perspective? Recent advances in science, including the ability to categorize DNA sequences, have enabled scientists to accurately determine how closely species are related to each other. This has led to the revision of the original grouping suggested by Linnaeus. New organisms are constantly being discovered, and relationships between organisms are changing all the time, so the importance of a taxonomic study to modern science cannot be underestimated.